The special senses. The body's special senses are senses that have organs designed especially for them. The five special senses are taste, smell, sight, hearing, and equilibrium.
The tongue is an elaborate complex of heterogeneous tissues with taste organs of diverse embryonic origin. The lingual tastes are papillae, composed of an epithelium that includes specialized taste buds. The basal lumina and a lamina propria core with matrix molecules, fibroblasts, nerves, and bristles. So taste buds have very sensitive microscopic called microvilli. Those tiny hairs send message to the brain about how something tastes. So you know if it's sweet, salt, sour, or bitter. The average person has a 10,000 taste buds and they're replaced every 2 weeks or so.
The purpose of papillae. Papillae are the little bumps on the top of your tongue that help grab food while your teeth are chewing. They also have another special job. They contain your taste buds, the things that help you taste everything from sour lemons to sweet peaches. How can we taste sour, salty, bitter, or sweet taste? Well, humans can detect sweet, sour, salty, bitter, and savory taste. This allows us to determine if foods are safe or harmful to eat. Each taste is caused by chemical substances that stimulate receptors on our taste buds. Your sense of taste lets you enjoy different foods and cuisines. Which taste is most sensitive? The answer is bitter. Because bitter, of all the flavors, our taste buds are most sensitive to bitterness, with most people able to detect bitter flavors, even in every small quantities. This is because many toxic substances have a bitter flavor and humans have evolved to react quickly when it is detected. The chemical senses smell and taste. The receptor for smell, olfaction, and taste gustation are classified as chemoreceptors because they respond to chemicals and solution. The olfactory epithelium occupies an area of about 0.5 cm in the roof of each nasal cavity. The olfactory receptor cells are bipolar neurons whose olfactory hairs or cilia protrude from the epithelium. Axons emerging from their basal ends are gathered into small fascicles called olfactory filaments. These penetrate the cribriform plate of the ethmoid bone and proceed as the olfactory nerves. The synapse and the olfactory bulbs lying on either side of the crista galli of the ethmoid bone. Impulse from neurons of the olfactory bulbs are then conveyed to olfactory portion of the cortex. Smelling and the nose. Our sense of smell helps us decide what is safe and what is harmful. When we smell something good or pleasant, we decide it is safe to be around or safe to allow inside our body. When we smell something bad or unpleasant, we wrinkle our nose and move away from it or prevent it from entering our body. Smell comes from aroma molecules that float in the air and travel up our nostrils and into the nasal cavity, which is a large open space behind the nose. The aroma molecules that travel to the roof of the nasal cavity and stick the tiny little hairs called cilia. These cilia, which belong to smell receptors, send information about the smell to to the olfactory bulb, which forwards the information to different parts of the brain. When we sniff, we force more aroma molecules to enter our nose and bend to our smell receptors, so our brain has a better idea of what we are smelling. Our sense of smell is closely tied to the area in our brain that controls memory and emotion, which is why we can instantly recognize certain smells. Tasting flavors with our nose and tongue. Our nose and tongue work together when it comes to tasting flavors in food. In fact, much of the flavors we taste come from our nose and sense of smell. Our tongue is limited to detect only 5 tastes, but our nose can sense over 10,000 smells. The ability to smell food aromas is what allows us to taste complex varieties of flavors in different foods. It's no wonder why we can taste food when our nose is stuffy.
The human eye. Eyes are very similar to camera. Like cameras, eyes take pictures of the world around you. The human eye is a sense organ and a part of a sensory nervous system. The eye has many parts that must work together to produce clear vision. Now let us talk internal or the visible parts of our eyes and the internal of our eye. The sclera, the white part of the eye, protects the eyeball. It covers the entire outside of the eyeball except the cornea. Eyebrow, the prevention of sweat, water, and other debris from falling down into the eye socket. Eyelashes, the first line of defense for your eyes, keeping airborne dirt and dust from reaching the delicate eye tissue. Eyelid are responsible for protecting their surface against foreign elements and other stimuli that could damage them. In addition, each time we blink, our eyes receive a set of substance that moisten them and prevent them from drying. Pupil, to control the amount of light that allowed to enter the eyes. Iris, controlling and helps your eyes to see clearly. Now we are done talk about the visible. Now here we are, let's talk about the internal structure. The first one is sclera, the supporting wall of the eyeball. It helps maintain our eyeball's shape and protects it from injury. The choroid, the vascular layer of the eye, lying between retina and the sclera. It contains blood vessels that nourish the retina. Pupil, this is a sort of hole in the middle of your iris, or black dot at the center of the eye. It's an opening through which light can enter the eye. Size of the pupil decreasing or increasing depending upon the intensity of light entering the eye. Iris, the colored part of the eye that controls or regulates the amount of light that enters the eye. Cornea, the clear layer of the front of your eye, covers the iris and pupil that transmits and focuses light into the eye to make sure your eye records clear images. Main function is refract and bend the light. Lens, located behind the pupil, acts like a camera lens that focuses light rays onto the retina. It can also become thicker or thinner to make the pictures as clear as possible. It is made of a transparent jelly-like substance which is made of proteins. Retina, the sensory membrane lying at the back of your eye. It composed of several layers of cells called photoreceptors. There are two types of photoreceptor cells, the rods and cones. Rods process black and white, while cones process colors. These cells turn images into electrical impulses that send to the brain through the optic nerve. The optic nerve, the electrical impulses formed by the retina travel along the optic nerve to the brain. Basically, the optic nerve is a path that connects the eye and the brain. Vitreous body is a clear jelly-like substance that fills space between lens and retina. It helps to preserve eye shape.
Light passes into the eyes through the cornea, which is also called windows of the eyes, and then through anterior chamber, into the pupils, the round opening, in the center of the iris. The iris, which are a flat, colored, ring-shaped membrane behind the cornea of the eyes, may dilate or constrict to determine how much light gets to the lens. Then, the lens moves light through the clear cavity, called the vitreous, and focuses it on the retina. The retina then converts this light energy into electrical impulses, and passes it through the optic nerve, also called the second cranial nerve. These impulses then travel through the optic nerve into the primary visual cortex, which is located in the most posterior portion of the brain's occipital lobe. While it may sound like a lengthy process, but takes only about 13 milliseconds.
Pathway of light through the eye and light refraction. When light passes from one substance, another substance that has a different density, its speed changes and the rays are bent, or refracted. Light rays are bent in the eye as they encounter the cornea, the aqueous humor, lens, and the vitreous humor. The refractive bending power of the cornea and the humors is constant. However, that the lens can change by changing its shape, that is, by making it more or less convex, so that light can be properly focused on the retina. The greater the lens convexity bulge makes it thicker, the more it bends the light. And the flatter or thinner the lens, the less it bends the light. We can see from the figure A that the resting eye is set for distant vision. In general, light from a distant source over 20 feet away approaches the eye as parallel rays. And the lens does not need change shape to focus properly on the retina. However, light from a close object tends to scatter and diverge or spread out, and the lens must bulge more to make more closer vision possible, as what can we see in the figure B. To achieve the ciliary body contracts toward the lens, slackening the ciliary zonules and allowing the lens to become more convex. This ability of the eye to focus, especially for close objects, those less than 20 feet away, is called accommodation. The image formed on the retina is a result of the light bending activity of the lens is the real image. The normal eye is able to accommodate properly. However, vision problems occur when lens is too strong or over-converging, or too weak, or when there are structural problems of the eyeball. Axons carrying impulses from retina are bundled together at the posterior aspect of the eyeball and leave the back of the eye as optic nerve. At the optic chiasma, the fibers from the medial side of the brain of each eye cross over to the opposite side of the eye. The optic tract fiber synapse with neuron in the thalamus whose axons from the optical radiation, which runs to the optical lobe of the brain. There, they synapse with the cortical cells, and visual interpretation occurs. Each side of the brain receives visual input from both eyes, from lateral field of the eye on its own side, from the medial field of the other eye. Also notice that each eye sees a slightly different view, but their visual fields overlap quite a bit. As a result of these two phenomena, humans have binocular vision. Literally, two-eyed vision provides for depth perception, also called three-dimensional vision, as our visual cortex fuses the two slightly different images delivered by the two eyes in one picture. The eye that focuses the images correctly on the retina is said to have emetropia, literally a harmonious vision. Nearsightedness or myopia, also called a short vision, occurs when the parallel light rays from distant objects are focused in front of the retina. Nearby objects are in focus, however, because the lens accommodates to focus the image properly. Correction requires concave correction lenses that diverge the light rays before they enter the eye, so that they converge farther back on the retinal surface. Farsightedness or hyperopia occurs when the parallel light rays from distant objects are focused behind the retina. People with hyperopia see distant objects clearly because their ciliary muscles contract continuously to increase the light bending power of the lens, which moves the focal point towards into the retina. We also have astigmatism, unequal curvature in different parts of the cornea. In this condition, blurry images occur because points on the retina retina, but lines. Special cylindrically ground lenses or constructs are used to correct astigmatism, the internal muscles. These muscles include ciliary body, which alters lens curvature, and the radial and circular muscles of the iris, which control pupil size. The external muscles are the rectus and oblique muscles attached. They are also responsible for convergence, the reflective movement of the eyes, medially when we view close objects. When convergence occurs, both eyes are aimed toward the near object being viewed. When the eyes are suddenly exposed to bright light, pupils immediately constrict. This is called photopupillary reflex. It prevents bright light from damaging the delicate photoreceptors. The pupils also constrict reflectively when we view objects. Accommodation pupillary reflex provides acute vision.
Hearing is the process by which sound vibrations from the external environment are converted into action potentials. A vibrating object makes noise, like a guitar string, and this vibrating pressure pulsates on air molecules, more commonly known as sound waves. The sense of hearing is provided by receptors in the cochlear duct. They are hair cells similar to those of the semicircular canals in vestibule. However, their positioning within the cochlear duct and the organization of the surrounding structures protect them from stimuli generated by anything other than sound waves. The frequency and intensity are the waves stimulate the hair cells along the cochlear spiral. The frequency of the perceived sound is detected by the part of the cochlear duct that is stimulated. The intensity or volume of the sound is detected by the number of hair cells that are stimulated at a particular point in the cochlea. There are ducts within the bony labyrinth of the cochlea. First, the vestibular duct or scala vestibuli connects to the oval window. Next, the tympanic duct or scala tympani connects to the round window. Third, the cochlear duct or known as scala media is separated from the tympanic duct by the basilar membrane. The hearing process. Number 1. Sound waves travel down the external auditory canal and arrive at the tympanic membrane. 2. Movement is created in the tympanic membrane, which leads to the movement of the ossicles and the amplification of the movement. 3. Movement of the stapes at the oval window creates pressure waves in the perilymph of the vestibular duct. 4. The pressure waves distort the basilar membrane. The location of the maximum distortion depends on the frequency of the sound, as the basilar membrane varies in the width and flexibility along its length. Higher pitched sounds create maximum distortion near to the oval window and lower pitched sounds further away from the window. The amount of distortion gives sensory information as to the volume of the sound. Next, vibration in the basilar membrane leads to vibration of the hair cell cilia against the tectorial membrane, leading to the release of neurotransmitters by the hair cells. As hair cells are arranged in rows, a soft sound may only distort a few hair cells in a single row, but more cells in more rows will be stimulated as the volume increases. And the last one. Information about the region of stimulation and the intensity of that stimulation is relayed to the brain via the cochlear branch of the vestibulocochlear nerve or cranial nerve 8.
Equilibrium. Along with hearing, the inner ear is responsible for encoding information about equilibrium, the sense of balance, which it does in the vestibule and semicircular canals, structures that are sometimes collectively referred to as the vestibular apparatus. Several types of sensory receptors provide information to the brain for the maintenance of equilibrium. The eyes and the proprioceptors in joints, tendons, and muscles are important in informing the brain about equilibrium. However, unique receptors within the inner ear play a crucial role in the monitoring equilibrium. There are two types of equilibrium: static and dynamic equilibrium. Static or gravitational equilibrium, which involves the movement of the head with respect to gravity. Dynamic equilibrium or rotational equilibrium, which involves acceleration of the head in rotation, horizontal, and vertical movements. The perception of equilibrium occurs in the vestibular apparatus. Motion in the following two structures is detected as follows. The vestibule is the primary detector of changes in static equilibrium. A sensory receptor called a macula is located in the walls of the saccule and utricle, the two bulb-like sacs of the vestibule. A macula contains numerous receptor cells called hair cells, from which numerous stereocilia, long macrobuli, and a single kinocilium, a true cilium, extend into a glycoprotein gel, the otolithic membrane. Calcium carbonate crystals called otoliths pervade the otolithic membrane, increasing its density and thus its responsiveness to changes in motion. Changes in linear motion cause the otolithic membrane to move forward and backward in the utricle or up and down in the saccule. The movement of the otolithic membrane causes similar movements in the embedded stereocilia of the hair cells, which in turn initiate graded potentials. The semicircular canals are the primary detector of changes in dynamic equilibrium. The three canals, individually called the anterior, posterior, and lateral canals, are arranged at right angles to one another. The expanded base of each canal, called an ampulla, contains a sensory receptor or crista ampullaris. Like the maculae of the vestibule, each crista ampullaris contains numerous hair cells, whose stereocilia and canocilium protrude into a gelatinous matrix, the cupula, which is analogous to the otolithic membranes to the maculae. Changes in rotational motion cause the cupula and the embedded stereocilia to move, which stimulates the hair cells to generate a graded potential. Graded potentials in the hair cells of the maculae and cristae result in changes in the amounts of neurotransmitter secreted. In response to these changes, action potentials are generated in the fibers of the vestibular nerve, which subsequently joins the vestibulocochlear nerve. From here, the nerve impulses travel to the pons and the medulla oblongata. Other information about the special senses.